This lecture is on the cell cycle. We're going to talk about it specifically in respect to its role in cancer. The cell cycle can also be called cell division, and it is the process where one cell divides to become two identical cells. These two identical cells are now called daughter cells, and the cell you started with is the parent cell. Cell cycle, cell division, this happens all the time. It happens when we're growing up as children. It happens if we get a paper cut or some other injury to our body, so wound repair, replacing damaged and dead cells with new healthy cells. But it can also go wrong, and it can happen where the cell divides abnormally. And this can lead to cancer. So under normal conditions, non-cancerous conditions, cells that are exposed to the environment are replaced frequently. When I say exposed to the environment, obviously you're going to think of skin. And if you think about it, it makes sense. Your skin's exposed to UV radiation when you go out in the sun. It's exposed to anything that might be in the air that's a potentially harmful substance for your skin. But we also consider your digestive tract, the cells lining the digestive tract, as being exposed to the environment. In this case, the environment is food. And sometimes there can be harmful compounds that you ingest with your food. They can be part of the food or they can be on the food. So these cells, because they have a high potential for damage because of the environment they're exposed to, those cells are going to divide a lot. On the other end of the scale, nervous tissue, like your brain, your nerves, your spinal cord, those cells very seldom divide after early childhood. This is why nervous tissue damage, including brain damage, can be so devastating because these tissues don't divide to replace damaged cells. Now, the whole process of cell division is highly, highly regulated. And there are lots of ways it's regulated. We're going to start by talking about a couple of different ways that this is regulated. The first of which is the cell will not start dividing until it gets the correct signal. And this signal can be in the form of hormones. It can be triggered by nearby cells being damaged. But there will be a signal that says, go, start going through the cell division process. And then once the process starts, there are multiple checkpoints that make sure everything's going okay. So you can see on this slide, I've got the G1 checkpoint marked. The G1 checkpoint looks for, is the cell big enough to divide? Is there enough nutrition inside the cell to divide? Is the environment around the cell suitable? Is there space available? Most cells will not divide if they're pressed up against other cells. And there are multiple checkpoints throughout the process, and they check for different things at different times. The immune system also looks at the cell division process. And if it looks like something's going wrong, your immune system will often come in and kill that cell or trigger a process where the cell kills itself. We're going to talk about that process called apoptosis in a later lecture. So here we have on the left healthy, an x-ray of a healthy non-cancerous person. You can see the heart, you can see the liver, that's most of this white part at the bottom. You can see faint outlines of the clavicles, the ribs, and you can see this dark space, which is the lungs. The lungs don't show up very strongly on x-rays. Some of this other stuff is things like blood vessels. On the right-hand side, however, there's a mass in the lung of this person. And you can see this opaque, solid structure right here. Cancer is disruption of the cell cycle when cells divide out of control. Most cells, most human cells, have a limit on how often they can divide, and that limit is around 50. Cancer cells do not have this limit. They're often called immortal cells. They can just keep dividing and keep dividing and keep dividing. It makes them very, very useful in medical research. So, what causes cancer? Let's talk about the genetics here. Mutations in your genetic material, changes in the DNA, are changes in the information of the cell. Most mutations do not have an effect on the cell at all. You know, if you've got a mutation in a liver enzyme in a cell on your skin, that doesn't matter. You're not making liver enzymes anyway on your skin. If there's a mutation in a gene involved in hair color in your liver, it doesn't matter. You're not growing hair in your liver. So the mutations have to be in certain genes. One mutation is not really enough to trigger cancer. You need multiple mutations. And it can take years or decades from a cell with a single mutated gene to get more mutations and grow into a cancer that is large enough to be detected. But again, most genetic mutations do not result in cancer. Like I said, it has to be in specific genes. These types of genes are called proto-oncogenes or tumor suppressor genes. And we will talk about those in just a second. Few more quick things to say before we get on to the proto-oncogenes. Your cells have 2 copies of each gene, except for males. And in males, you have an X chromosome and a Y chromosome, and you don't have a second copy. But you have second copies of all the other chromosomes, so you have second copies of their genes. And there are lots of backup systems to prevent the abnormal, harmful growth of cancer. So let's talk about the genes involved here. We're going to talk about proto-oncogenes. Proto-oncogenes are involved in promoting cell division. These are the proteins that say, yes, it is time for this cell to divide. They turn on the cell cycle. when there are mutations in these proto-oncogenes, they get stuck in the on position. Now we call them oncogenes. And it's like you're pressing down on the gas pedal and not lifting your foot up, and you're really going with that foot all the way down, trying to drive as fast as possible. And the cells just start dividing and keep dividing and keep dividing. An example of this is BRCA1, BRCA1, breast cancer predisposition gene number 1. Mutations in BRCA1 increase the risk of breast cancer and ovarian cancer. Now, it's increasing the risk, and that's something you need to keep in mind. A mutation in BRCA1 is not a guarantee of breast cancer, but it does increase the risk of breast cancer. On the other side, there are genes called tumor suppressor genes, which do exactly what you think. They suppress cell division. These are off switches or brakes. If these are mutated, it's like you're driving a car that doesn't have brakes. Your proto-oncogene, the gas, is pushing towards cell division, but you can't stop it. And so again, you get multiple rounds of cell division. One of the things tumor suppressors do is promote apoptosis. Apoptosis is also called programmed cell death, and it is a very tightly controlled mechanism where a cell that is abnormal, it's on its path to becoming cancerous, or it's infected with bacteria, that cell kills itself and stops the process right there, right? When these tumor suppressor genes are mutated, the cell can no longer stop abnormal cell divisions. One of the most important tumor suppressor genes is a gene called P53. Over half of cancers have mutations in P53. P53 looks for structural changes in the DNA. So the DNA in your nucleus, it goes along and it looks for things like breaks in the DNA that shouldn't be there. It doesn't look for all mutations. It's not looking at changes in information. It's just looking at physical defects. And it can repair lots of these. And if it doesn't repair them or can't repair them, then it triggers apoptosis. Okay, so that kind of wraps up the cell cycle and the role of the cell cycle in cancer.